Hey everybody, we're going to continue on talking about evolution. So the first question we need to look at is the unit of evolution the individual or the population? And the answer is that while evolution affects individuals, it can actually only be tracked through populations. And so we need to study populations and not individuals. We also need a method to track how populations change over time, and we do that through a study called, or through an area of study called population genetics. And so this is when we study, again, genetic variation within a population, and this is sort of where we take what we learned with Mendel and mix it together with what we're talking about with Darwin. So population genetics uses how we look at alleles and genes to track and study evolution over time. And so we're looking at the genetic basis for the variation that we can see. So for the phenotypic variation that we can see and how it interacts with the environment and how natural selection can select for or against certain traits. So we want to review really quickly the sources of variation from sexual reproduction. You've got random assortment of chromosomes. So the way that chromosomes line up during meiosis is random. Random fertilization is which egg is fertilized by which sperm is completely random. And then you also have crossing over, which happens again during meiosis, and it's when you've got little pieces of homologous chromosomes that break off and switch places with each other. And then you also have mutations. Now mutations are inherited changes in a gene, and rates of mutation are usually really low because the DNA repair mechanisms work so well. So a population is going to be a localized group of individuals of the same species. And so when we, again, are looking at evolution, we're not looking at individuals and we're not looking at a whole species. We are looking at one group of individuals of the same species who live in the same area. Okay. Now a species is larger than that. A species is going to be a group of similar organisms. They usually have multiple populations, and multiple populations are considered the same species if they could potentially interbreed and produce fertile offspring. Now we're also going to be talking a lot about genes and alleles, and we're going to be referring to them as the gene pool. So the gene pool is the total aggregate of genes within a population. So if evolution is occurring, then you must see changes in the gene pool over time. So if we're seeing phenotypic changes and actually able to observe evolution, then we should also be able to see differences in the allele frequencies within that population as well. And when we look at those allele frequencies in the gene pool and any changes that may be occurring, we call that microevolution. Now to track all of this and to measure if evolution is occurring, we use what's called the Hardy-Weinberg theorem. Now this was developed in 1908, and it's a mathematical model of gene pool changes over time. And it asks the question, does sexual reproduction by itself cause changes in gene frequency? Now there is a basic equation where we say P plus Q equals 1, when P is the percent or frequency of the dominant allele, and Q is the percent or frequency of the recessive allele. And so if you only have two alleles for a given gene, then P plus Q should equal 1 because you've got the dominant, you've got the recessive. When you add them all up, you should get 1. Then you have the expanded equation. And this is when you say P plus Q squared equals 1 squared. And when you factor it out, you find out that P squared plus 2PQ plus Q squared equals 1. Okay, now in this case, the homozygous dominance would be considered that P squared because P is for the dominant, and when you are homozygous dominant, so let's say we're talking about height in plants, you would have capital T for a homozygous dominant, so it's like two T's together, and P squared is multiplying two P's together. With the heterozygote, you would have 2PQ, and so again, if we're looking at plant height and we've got T's, a heterozygote would be capital T lowercase t, and then the homozygous recessive is going to be Q squared, because if you're looking at a homozygous short plant, they would have two little t's, and when you look at Q squared, you've got two Q's that you're multiplying together. So we're going to look at an example population. We're going to focus on flower colors. Capital A is going to be red flowers, lowercase a is going to be white flowers. So real quick before we start looking at all the math, this population has 500 total plants. 320 of them are homozygous dominant for red. 160 are going to be heterozygous for red, and then 20 will be homozygous recessive for white. So if we're starting with a population of 500 plants, And we want to look at genes first. Okay, so we're looking at alleles. If we've got 500 plants, we know that 480 of them have the genes for red. And we know that because we've got 320 of them are homozygous dominant for red and 160 are heterozygous for red. So we've got 500 plants, 480 of them are going to be red, and then 20 of them will be white because they are homozygous recessive. So now that's 500 total plants, but because each plant has 2 alleles, that gives us 1,000 alleles or genes total within this population. So when we want to calculate P, which is the percent of the dominant allele, what we're going to look for is the number of individuals who are homozygous dominant. So in this case, 320. Multiply that by 2 because these 320 individuals all have 2 copies of that allele. And then we have 160 individuals who are heterozygous. And so when you do 320 times 2 plus 160, it gives you 800. So 800 out of the 1,000, or 80% of the alleles within the population are dominant. When you look at recessive now, you take the other 160 because 160 of the individuals are heterozygous, and so they all have one recessive A, plus the 20 individuals who are homozygous recessive, and they have 2 recessive A's, so you'll do 20 times 2, and you add those together, it gives you 200 individuals out of 1,000, which is 0.2, or 20% of the alleles within this population are recessive. All right, and so now when we expand this equation out and we sub in for P and Q, we found out that P is 0.8, we found out that Q is 0.2, so we've got P squared plus 2PQ plus Q squared. So P squared would be 0.64, 2PQ would be 0.32, and then Q squared is 0.04, and that equals out to 1. Okay, we can also do this a different way, so we can look if we're counting up the dominant alleles. So P squared is 0.64. When we look at P and Q, here we're only looking at 1 PQ instead of 2 PQ because, again, for the heterozygous, we're only looking at half of their alleles, gives us 0.16, and so again that shows us 80%, and we can do the same thing for the recessive allele. And so what this showed us with this flower population is that the gene pool is in a state of equilibrium. It has not changed because of sexual reproduction, so no evolution has occurred here. Now, Hardy-Weinberg is important because it's a yardstick that's used to measure rates of evolution. And so Hardy-Weinberg actually predicts that gene frequency should not change over time as long as the Hardy-Weinberg assumptions hold. Okay, so it says no evolution should occur. And we can use Hardy-Weinberg to calculate gene frequencies through time. Now, if we've run this a couple times and we see that gene frequencies are changing, then we know that evolution is occurring. Another example would be looking at the PKU allele, which is expressed in an individual that is homozygous recessive. So we know that PKU is found in 1 out of 10,000 births. We also know that, as I said, it is a homozygous recessive condition. All right, so what that tells us is that 1 out of 10,000 individuals has Q squared, and so Q in this case equals 0.01, which means P would equal 0.99. Now, so when we expand this equation out, we can see that when you do the 0.99 squared plus 2PQ plus Q squared, you get 1. And so you would want to expect 98% of individuals to be normal, about 2% to be carriers, and then the 0.01% to actually have PKU. So when you are doing AP problems using Hardy-Weinberg, you need to pay attention to what you are being asked for, okay? So if you have, and what you're being asked to solve for. So if you're being asked to solve for Q squared versus Q, if you're being asked to solve for P, like, you just have to make sure that you're reading the problems correctly and that you understand the information that you're given and you understand what you're being asked to solve. Now, the other thing, this is not completely correct. So calculators were not allowed prior, on the prior AP exam. You can have calculators, but they are simple four-function calculators, all right? But Hardy-Weinberg is pretty much always on the National AP Bio exam, so just make sure you're ready for it. Now, the Hardy-Weinberg assumptions that I talked about before was that you have to assume that populations are large. You have to assume that they are isolated so that you don't have individuals migrating in and out. You want to assume that there is no net mutations. You want to assume that mating is random and that there is no natural selection acting. So those are the 5 assumptions of Hardy-Weinberg. It says that evolution is not occurring if the population is large, if it is isolated, if there's no net mutations, if all mating is random, and if no natural selection is occurring. All right, so the gene frequencies will not change over time and evolution will not occur if those 5 things are true. So now your lecture question for today is, how likely will natural populations hold to the Hardy-Weinberg assumption? So do you think that it's very likely that populations would exhibit those 5 assumptions for a long period of time, for a short period of time? Tell me what you think. Please send me a message via the website. Thanks, guys.